statistical models and mechanistic computer models. So statistical models, and here I mean both traditional statistical models like linear regression, but also the modern machine learning models, AI models. So these statistical models, they capture the patterns between inputs and outputs, and then you train them either using deterministic methods, like that's the case of traditional models, or stochastic methods. That's the case of AI, for example, deep learning, deep network models. And then this allows you, if you capture the statistical relationships in the training data, this allows you to predict new outputs also for untrained data. But in contrast, and my talk will be mostly about the second kind of models, mechanistic models. They are different because they not only capture statistical patterns in the data, but here we have a bigger ambition because we also want to understand the system. So that's why we then implement in these mechanistic models anatomy, physiology, biophysics, or biochemistry sometimes. And maybe you are familiar with these models. So for example, here you can see a nerve cell, which you can represent as an RC circuit. Why is this so? Because a membrane of the neuron can be represented as a capacitor, because to change the voltage across the membrane, you have to distribute the charge, and this is basically a capacitor. And then the ion channels can be represented as conductances. And then so if you are familiar with compartmental modeling, then you know about this. If not, then this is a very quick summary. So you can use Ohm's law to describe currents that flow through those channels, and you can use capacitance equation to describe the capacitance properties of the membrane. And then if you combine those equations, you can get very complex mechanistic models. Here, just very briefly, I want to mention that you can combine this classical compartmental modeling, Hodgkin-Huxley type modeling, with the volume conductor theory. And this has been well established in the past years, mainly by the group of Gaute, Einevol. And if you combine these multi-compartment models, MC, with volume conductor theory, then you can also model in a very rigorous way also extracellular electric signals like LFPs, EEGs. And then also you can make it also in a different order. So you can first model electric fields, and you can couple them to multi-compartmental models. So that's what we also started doing. I don't want to go into details here. This is just to let you know that this is possible. You can simulate electric fields and couple them to those biophysically detailed or also morphologically detailed biophysical models. Statistical models, AI models, and mechanistic models can be also combined very productively and synergistically. One example for this is SBI, or simulation-based inference, which allows you to identify those models, those mechanistic models, that generate simulations that are consistent with experimental data. And this has been mostly established in the lab of Jakob Macke. So they have this artificial neural network that can learn to predict the distribution of parameters. So you see here a prior distribution of parameters for any model. It can be it can be applied also to other models, not only to neuronal models or biophysical models, but here we have an example for a biophysical one. So you see here an RC circuit and electrophysiological data, and you can then use it for this artificial neural network to basically determine the a posteriori distribution of parameters for your ion channels, for example. And then you know that if you take the distribution that this neural network learned, then you can, without running the simulation, you can basically generate very realistic, let's say, voltage traces, for example. So it's good to know about this method. So I won't go into details here, but this is one example how AI models can basically improve mechanistic models. And here, again, without going into details here, I just want to mention this and give you one reference. So we applied it, for example, to models of synaptic plasticity. So if you are interested in models of synaptic plasticity, but also in other models, you can use SBI to find the distribution of parameters that is consistent with data. So here you see, I don't have to explain all the details, but you see that the model, which is this dashed line, is consistent with experimental data. And in this case, this was plasticity data. And here we simulated the plasticity in hippocampal cells, and the strength of the synapses, or the spine size, the size of the spine, is a proxy for synaptic weight. So one example in which we used SBI. And then also, again, without going to details too much, you can do this also in the opposite direction. So you can first look into mechanistic models, and you can improve AI models. So this is one example. So sometimes people call this neuro AI, right? So you can take something that you learned in biology, for example, some dendritic mechanism. So here we took what we call dendritic normalization mechanism. I will come to that later probably. And then we implemented it in the network. And then this network with this dendritic mechanism, that's this orange curve, it learned better than a network without the dendritic mechanism. One example in which neuroscience knowledge improved the learning of the artificial neural network. Okay, so that was just a very, very general introduction or remarks. So now I would like to talk about the methods for creating robust mechanistic models and also morphological models of nerve cells. Here, so if you are not familiar with compartmental modeling, this is a very brief recap of what I already mentioned. So to model a neuron, so here I don't mean necessarily a robust model. So if you want to create a model, then this is the classical Hodgkin-Huxley type of modeling, right? You need three equations. You need, as I mentioned, Ohm's law, capacitance equation, then also the law of the conservation of charge and current. And if you combine them, then you get the famous Hodgkin-Huxley equations. But we don't have to go into this because this is textbook knowledge in computational neuroscience. So there is a problem because we need more robust and more reliable biophysical models of neurons. Why this is so? Because typically people have been using different morphologies, or the models, you tune them to one data set, but then they fail for another data set, or models use a mixture of ion channels from different species, or they cannot account for species differences. So these biophysically complex models have some issues, and they have been nicely summarized in this paper by Almog and Korngreen 10 years ago. So if you want to read about the issues of realistic neuronal modeling, that's a nice paper to look at. But the question is, how are we going to address these issues? So it's interesting that real neurons are very diverse, right? We know that they are diverse in morphology, but they are also very diverse and heterogeneous also regarding their ion channel properties. So they have multiple ion channels with different densities. But despite of this diversity, they work very well. So they are robust. But our models are not. So the question is, how can we make our models more similar to real neurons? Because then the models will have a higher predictive power. And so how we address this issue? So together with my colleague Hermann Kunz, we combine the classical biophysical modeling with morphological modeling. I will explain to you later what that is. So we combine it. We have our own software, T2N, which stands for trees to neuron interface, because the trees toolbox allows you to create morphological models of nerve cells. And then we combine it with principles of optimality, because we think that if you ask what would be the optimal way to implement a function, this can also then improve your models, because very likely real neurons are also optimal for several objectives. So this normative approach is, I think, very helpful. Here I'm going to give you one example for a morphologically robust model of hippocampal granule cells. We have created this back in 2017, but we still keep using it. Here you see the 3D morphology of these cells, because you need 3D reconstructions to create morphologically complex models, because they will then change the conductances in the model, right? So morphology matters. And then we equipped those morphologies with 15 voltage and calcium-dependent channels. We don't have to go into details here, but Marcel Beining, who created this model, was very careful. And he read a lot of papers, and he read papers about the distribution of these ion channels. And so we think that this is one reason why this model is so robust. So you can see here, we don't have to go into all these details, but here on the left, you see the expression of different ion channels. So the thickness of the line stands for the density. And you see that different channels are expressed in different parts of the dendritic tree of this hippocampal granule cells. So for example, you see a high expression of the NAV 1.6 channel in the axon initial segment and in the axon, but no expression in the dendrites. And then the model was tuned to replicate patch clamp experiments. Again, we don't have to go into details here, but you see on the left the experimental data. Here are some IV relationships, so relationship between the current and the voltage. You see here also one curve, barium. So you can run simulations in silico for pharmacology, right? You can block some channels, and if you know that barium blocks some channels, this can change the current voltage relationship, and the model was able to replicate this. So we replicated several data sets. Here you see only examples for the number of action potentials. Then you see here blue line. So this is biophysical modeling in reconstructed morphologies. So we inserted those 15 channels into reconstructed morphologies. But you see here also simulations with green curves. So here we inserted the channels into synthetic morphologies. I have not explained to you how we generated them, but I will very briefly. So yeah, you have to be patient there. The nice thing was that the mouse model can be converted into a red model using a simple shift in the expression of KIR channels. So again, we don't have to talk about the details, but I think this is one important message, that if you want to create a model, it's good to replicate several data sets and potentially even species differences. And one more detail, just focus on this one panel. You don't have to look at all those panels. So if we change the ion channels according to the literature from immature granule cells, then we can also replicate the input-output curve, not only for the mature granule cells, but also for the immature ones, for the young ones. And it's known in the field of neurogenesis, or in the field of adult-born granule cells, that these young cells are more excitable. So you see here a leftward shift in this input-output curve. I'm mentioning this because this will be important also in the later part of the talk. Okay, so let me summarize. So this was one example for a morphologically robust granule cell model, because it can replicate electrophysiology across a wide range of morphologies. So why was the model robust? We don't know exactly, because I think there are still open questions. But I think one important point was that we used many morphologies for tuning, and we tried to reproduce more than one data set. And I think this constrains the model better and makes it more robust. Then also, the model replicated the species differences, which is another important point. And then, okay, this is a specific point that we were able also to replicate some properties of these ABGCs, which are adult-born granule cells. And as I mentioned, this will be important for the topic of Pareto optimality that we will come to later. Okay, so if you want to use this model, you can download it in the ModelDB, but so nowadays people share their models and their code typically, and that's important. Here, just very, very briefly, I want to show you another model that we created for CA1 pyramidal cells, but this model, as you can see here, has a simplified morphology. So this is not a full morphology, but the nice part is that, again, it replicates not only one data set, but 5 different experimental tests, which were included in the so-called HIPPO unit. That's a battery of tests that were established by, so in this paper, by a Hungarian group. And so we took those 5 tests and we validated this model with these 5 tests. Otherwise, I think we don't have to go into details here. Here you see those five different tests. It's 6 because you can do it for strong or weak action potential, backpropagation. But yeah, I think we don't have to go into details here. The main thing is that it's important to use many different data sets and tests. Here, actually, there is one caveat, because so far, I told you that we used for the granule cells many different morphologies, but we had only basically one ion channel composition. So the densities of ion channels were not very heterogeneous. So we found basically in this old 2017 paper, and also then for the CA1 pyramidal cell paper, only one possible solution in the complex biophysical parameter space. So keep this in mind, because we will return to this. And we will talk about so-called population-based modeling, or some people call it ensemble modeling or database modeling. So it's good to generate not one solution, but more than one solutions for the biophysical space. Okay, the take-home message for this part of the talk would be that it is very useful to use multiple standardized tests or benchmarks for models of canonical neurons and circuits. And this should, so if you use those tests, then it should increase the robustness of your models. Okay, now I'm returning to this topic of morphological modeling, because I didn't tell you how we generated synthetic morphologies. And now I will tell you how we did that. So this is basically the work of Hermann Kunz, who has been focusing on this topic for many years. So we had these synthetic morphologies very briefly, and in a very simplified manner. You can say that you distribute target points in the 3D space, then you connect them, and then you get realistic dendrites. That's basically morphological modeling. The important part is that the real morphologies are very similar to synthetic morphologies, right? You can see here synthetic morphologies. In the case of granule cells here, quantitatively, basically mimicked the reconstructed real morphologies for different features like dendritic length, thickness of soma or dendrite diameter, or the number of branch points, etc. So, okay, but this sounds like magic, right? You distribute points, and then you generate synthetic dendrites, and then they are very similar. It's not magic. It is based on this algorithm that Hermann developed several years ago. And it's a very simple and elegant model, because you distribute points and then you connect those points, but you are trying to minimize 2 costs. One is the overall cable length, and the other one is the path length. And the nice thing is that you have one free parameter, the balancing factor, BF, and you decide what is more important. So if you say you want to minimize the path length cost, you can see this also as a conduction delay. So if the path length from the tip of the dendrite towards the soma is the shortest possible path length, then this is also good for the propagation of signals, right, from the distal parts of the dendritic tree towards soma. So you can see this also as a conduction delay. Or you care about the overall total cable length, and you can see this also as energy, right, because cable length costs a lot of energy. So if you minimize cable length and you don't care so much about minimizing the path length, then you get morphologies like this. But then the conduction delay or the signal propagation is not very efficient because you have to go from one part of the cell to the other part of the cell. Okay, but if you combine those two, then you can get very realistic and nice dendrites. You have to decide what is the balancing factor for different cell types. So that's the tuning of the model then. And also distributing the target points is not trivial. Okay, so here I want to show you that if you use morphologically diverse dendrites, that's very useful because this allows you to find general principles that are sometimes hidden behind the complexity of data. Again, without going too much into detail here, this is one example or one result that we got. And here we used, in this case, reconstructed morphologies, but then also in other panels of the paper also synthetic morphologies. But here in this figure, these are real morphologies downloaded from the neuromorpho databases. And so what we discovered that under some conditions, if you keep the passive properties the same, and then if you stimulate these neuronal models with synaptic stimulation, then the response of these neurons is largely independent of their size and the complexity of their dendritic tree. So you see here, yellow neurons are very small, and these red neurons are very large, but their voltage response at the soma is largely very similar. So here, we showed this for passive simulations. Then surprisingly, that was also the case for active models in which you also had some spikes, even including dendritic spikes. So here you see basically the same plot, right? The x-axis is the total dendritic length. The y-axis is the response, somatic firing rate. And this was the case also for active models, active CA1 pyramidal cell models. Yeah, so the synaptically driven somatic responses were, again, largely independent of the size of the dendrite and also of the branching. We call this finding dendritic constancy, and yeah, it is about the size invariance of synaptically driven responses. Interestingly, I showed you that the spike numbers are largely independent of the size, but not spike times. So the temporal neuronal code is still very related to the complexity of the morphology. But the spike numbers were pretty independent of the dendritic length and branching. So we see this principle as a backbone, as a passive backbone for neurons, which makes their somatic responses more robust. And here I mean spike numbers, not spike times. But of course, the cells are not that boring, right? It's not the case that all cells respond with similar firing rate. They can use this backbone for their cell type specific computations and plasticity. Okay, so now I am coming to the two key points or key words in the title of my talk, and that's degeneracy and Pareto optimality. So what does that mean? So if you are not familiar with this term, then I'm going to explain this, because Sometimes people confuse it with degeneration, and then they think, okay, this is the Alzheimer disease part of the talk. So it is not, okay? Please don't confuse this, the degeneracy with degeneration. So what is degeneracy? So I like the definition, and it has been nicely illustrated in this paper from Jean-Marc and from Yves Marder. Okay, so what is degeneracy? It is something similar to redundancy, but there is one difference. So redundancy means that you have identical components, let's say x and x prime, that contribute to the same function A, right? Degeneracy means that you have different components, let's say different ion channels, X and Y, that contribute to the same function, let's say similar somatic firing output. You can also have the case of pleiotropy. So one mechanism, one ion channel, for example, can contribute to two different functions. And typically in biology, you have a combination of pleiotropy and degeneracy, which we could call, or Yves Marder called this functional overlap. So that's a combination of degeneracy and pleiotropy. A classical example is the ion channel degeneracy. So if you are familiar with ion channels, then you know that there is a large overlap in the voltage activation and inactivation of these channels. So there is a large overlap in the voltage dependence domain. But there is also a large overlap in the time domain. So also the kinetics of these channels is not, it's not strictly separate in time. So there, again, the speed of their activation and inactivation is very overlapping. And that means naturally that those channels will contribute to similar electrophysiological patterns. So we have the definition, right? The degeneracy is the ability of different configurations of different components to perform the same function or to achieve or yield the same result. You can also see it as a many-to-one mapping between the parameter space or the trait space. So this would be, for example, ion channel space and the performance space. This could be the space of functions that you or neurons care about. By the way, a classical example for degeneracy would be the genetic code, because you have different triplets codons giving you the same amino acid, right? So that would be an example in the genetics. So this is one example from our own work, but we were not the first ones to discover this, because this has been discovered I think first by Astrid Prinz and Yves Marder, but we saw this also in our granule cell model. So I introduced to you the granule cell model, and I told you that there was this one caveat, right? That we basically had one solution in the biophysical space. We had multiple morphologies, but we have only one biophysical profile. So then later we took this model of Marcel binding, and then we varied stochastically the parameters for ion channels. You can see it here. So this y-axis, but now it's turned. This represents the expression of different ion channels. So you see here again these 15 different ion channels, and 0 means that the channel is missing, and 2 means a twofold increase in the expression. In modeling, that would be the maximum conductance of the channel. So twofold increase with respect to what? With respect to the original model from Marcel binding, okay? So we varied the conductance, and different colors represent different biophysical configurations of these ion channels. So basically different models, different biophysical models, but you see that they all generate very similar somatic firing patterns. So you have a many-to-one mapping between the parameter space and electrophysiological space. Here is one example. This is degeneracy you can find everywhere. So here, this is one example from heart cells. Here the action potentials look different, so they are much wider than neuronal action potentials. But you see in red the recordings, in blue the realistic models, and the black models are the invalid models. So they are not, the black models are all models, and some of them are realistic. So you see here many-to-one, or you could also say many-to-many mapping, because real cells also have heterogeneous responses. And so that's an example from modeling from the group of Blanca Rodriguez. And the nice thing is that then you can also simulate pharmacology in silico. You can block some channels, and you can simulate the widening of the action potential if you apply, for example, dofetilide in silico. Okay. One side remark is that you could apply these concepts also to pathology, right? So you could see a system, for example, a circuit in an animal or in human, in a patient, that would go from the healthy space into pathological space. And maybe it is the case that there are multiple routes or multiple paths to get from healthy state to similar pathological state. So there you can see this many-to-one mapping also, not only in the healthy context, but also in the disease context. And if you are interested in this topic, we wrote this up in a paper in which we talked about degeneracy in epilepsy, about multiple routes to hyperexcitable brain circuits. So if you're interested in this, you can check this out. Okay, back to our topic of creating robust models. So we observed this in our granular cell model, and even for 9-channel and 5-channel model, we were able to generate very similar somatic firing, right? So you could ask, okay, if you can achieve this with 5 channels, why do real cells use more than 5 channels? Why would they do this? So yeah, this is the case of ion channel degeneracy. So one answer would be that this diversity of ion channels allows a model neuron to better find the valid performance space. I'm not showing this here, so that's in another panel. But what I want to show here, that models, these 15-channel models, were more robust than 5-channel models, and that's maybe not so surprising. So if you remove a model, if you remove ion channels from these models, so from this 15-channel version, so let's say you remove, so this is minus CaV22, so you remove a calcium channel, or you remove a BK, a big conductance potassium channel, you can still get very similar somatic firing. And this is not the case for the 9 or 5-channel model, so they are more fragile. Okay, so the diversity increases the robustness of models, but also of real neurons. That's what we think, and also others. So now I want to go to the next topic. So this is a shift from degeneracy to the topic of Pareto optimality. And why we are shifting there? So now we are adding one more objective to our granular cell model, right? So now we care not only about somatic firing or about action potential backpropagation or about IV relationships. Now we care also about the biological function of these neurons. And we know from experimental literature that dentate gyrus is important for so-called pattern separation. So that would mean it is related to learning and memory. So let's say you hear my talk today, and you will remember, okay, Peter talked about degeneracy and Pareto optimality, and you will save this talk in your hippocampus. But then maybe you will hear my talk again in some other session or in some other workshop, or then for the third time. And now you saved the pattern, but you maybe will not remember the differences between the first, second, and third talk. So, or if you remember it, that would be the pattern separation. Or if you think about the rat in the room running around and navigating, then granule cells, they code for space, so they are also known to be place cells, but they also, so people think that they help the rat or also the humans to distinguish between different rooms that are very similar, right? So you can have a room that is similar to another room, and pattern separation would mean that you can separate those similar patterns. Now, okay, now we'll make a big jump from behavior to single cell modeling. I know it's a huge jump, but still, believe me that you can meaningfully represent pattern separation also at the level of spikes. So here we basically feed input spikes into our granule cell models, and then we measure output spikes, and the cell is supposed to separate them, right? So the cell is supposed to make the input spike patterns more dissimilar. Basically, if you inject noise, or if you delete some spikes, that already would introduce heterogeneity, and this would increase the pattern separation. However, because if you are deleting spikes, you also are deleting information. That's why we came up with our own measure of pattern separation, and we wrote our own pattern separation toolbox, which was the work of Alex Bird. So if you are in your projects, maybe, so some people need pattern separation, for example, if they study cerebellum or mushroom body or something else. If you want to quantify pattern separation, this might be useful because you don't want to confuse pattern separation with pattern destruction, right? Because then your cell is losing information. So Alex came up with these different measures of pattern separation based on information theory. And again, we cannot go into details here, and only Alex could explain to you all the technicalities. But the main idea is that you basically penalize the loss of information. So if you increase the sparsity, you delete spikes, that's good. You make the patterns more dissimilar, the input-output patterns. But if you lose information, then you also decrease pattern separation. So basically, it's a combination of sparsity and mutual information. Okay, you don't have to understand this in detail. I didn't even really explain this to you. So if you want to know the details, read Alex's paper. But just believe me now that we have a good measure to quantify pattern separation in our granule cell models, okay? Okay, so now, right, so basically we simulate temporal pattern separation, so separation in time, and that's what I mentioned. We have our pattern separation toolbox. So what we did is that we took data from experiments from Antoine Madar and colleagues. They actually recorded, or they fed input patterns into real granule cells in slices, and they recorded the output spikes. And so that was an experimental pattern separation paper. And then we took those data and we basically fed them into our pattern separation toolbox. And so we treated both the modeling data and also the experimental data the same, right? So we used pattern separation toolbox to quantify pattern separations. That's the y-axis here. We did that for different input correlations. And then we basically tuned the models, or we observed that our models were able to replicate real pattern separation of real granule cells. Okay, so this is just to make sure that our models can do that. But now the important question is the following one. The real granule cells do not reach the maximum pattern separation. I mean, we don't even know what's the maximum one, but like in principle, you could go higher, but they don't. And the question is why? So are they maybe suboptimal for pattern separation? That would be maybe surprising, right? If people think that those cells are specialized for pattern separation. And now we are coming to the end of my talk, and that's the topic of the Pareto optimality. So you can just replace the word Pareto with multi-objective, right? So we are now talking about multi-objective optimality. That means that we don't care about one objective, we care about more than one objective. In our case here, in cranial cells, we care, at least in this project, we cared about two objectives. One was pattern separation, quantified using the pattern separation toolbox and information theory, and the second one was energy efficiency. What does that mean? You can take those models, those mechanistic models, and you can basically count ionic flow for sodium and calcium, when you can convert it into ATP consumption based on physiology textbook knowledge, because in neurons you have pumps, the sodium potassium ATPase, which has to pump the sodium out of the cell, and it has to the pump has to maintain the concentration of ions. And this is a pretty established method how to estimate the energy consumption of a compartmental model. So we have two objectives. One is pattern separation, the other one is energy efficiency. And the interesting thing is that if you generate a population of models and they all can replicate somatic firing, however, they are not all equally optimal for the combination of these two objectives, right? So you see here a line, which we call the Pareto front. So this dark blue population of models was activated with 10 Hertz input, the other one with 20 Hertz input. But what we saw is that our original model, well validated by different experimental data sets created by Marcel Beining, was very close to what you could call a Pareto optimality for two objectives. Y-axis is the energy efficiency, so the higher you go, the more energy efficient you get, because the less ATP you consume or your model consumes, and the more to the right you go, the higher the mutual information or the better the pattern separation. Okay. Yeah. So, and what is Pareto optimality? Pareto optimality means that you cannot improve the performance of your model for one objective without degrading the performance for another objective. Okay, so we are not the first ones to use this. So I think one of the first scientists who applied this in biology was Uri Alon. And he has a very nice online lecture, but also an online chapter from a great book. So this is a great book. And there he explains the Pareto optimality in biology. He uses very simple examples. So for example, a car can be optimal for acceleration, but then it is very expensive, or it's very cheap, but then it cannot accelerate very well. According to Pareto optimality, all these cars would be equally optimal for the combination of the two objectives, right? But all the cars that would be below this Pareto front would be suboptimal. And the idea is that evolution removes these suboptimal points from the performance space and from the parameter space. Okay. Interestingly, this paper suggests that also dendritic morphologies might be Pareto optimal. And interestingly, these authors think that they are optimal for the two costs that Hermann uses for his morphological modeling algorithm, right? So here, x-axis is the wiring cost, so that would be the total dendritic length. The y-axis is a conduction delay, that would be the path length, and they seem to occupy the Pareto front, but this is still not completely nailed down, and because in this paper they did not generate the control group of morphologies very carefully, so this still has to be clarified, but it looks like even the morphology might be Pareto optimal for these two costs. Okay, now one last point that I would like to make is that is basically the link between degeneracy and Pareto optimality, because you saw that there is a huge degeneracy in our models, right? There is also degeneracy in real neurons, because that has been shown, for example, by Yves Marder. So also real neurons have different combinations of ion channels which generate the same pyloric rhythm in the stomatogastric ganglion of the lobster. That's the system that Eve Mather studied. So even real neurons are degenerate or exhibit degeneracy. However, the models that they're, you might ask, okay, from the huge theoretically possible parameter space, which ion channel combinations or which configurations actually exist in real neurons? And it's not so clear. And so we were suggesting in this paper that if we combine Pareto optimality with degeneracy, that this might maybe help. Why? Because if you have, let's say, parameter space, which is very complex, here we show it only schematically for 2 parameters, but you can think of 3 or more parameters. I think it's plausible based on Uri Alon's ideas that evolution over millions of years removed the suboptimal parameters from the parameter space, and it simplified the parameter space. So then maybe in reality you find only low dimensional manifolds in the parameter and in the performance space. So selection would be based on these trade-offs. And then what you would end up with would be neurons that would be close to the Pareto front, like the granule cells that we study, and they would be, they could be better in one task, and some of them might be even very bad for another task, or they would be good for a combination. We could call them generalists or specialists for task A or for task B. And maybe N6 and N4 neuron, maybe they don't even exist because evolution removed them from the parameter space, or from in this case, from the performance space. So that's the general idea. And this is, again, the definition of Pareto optimality, but you know that already. And so now I'm just summarizing, right? Because objective 1 was pattern separation. We showed that the models were able to replicate this. Then objective 2, energy efficiency. So real neurons, we think, are energy efficient. Maybe because of time, I will not go into detail here. This is about the mechanisms of energy efficiency, because this is linked to the overlap between the sodium and potassium current. But these are details. So if you are interested in that, we can discuss this later. But I think I will skip this. And I will skip that too. Here, very briefly, I'll show you that other cell types in the hippocampus, these red and violet dots, are CA1 pyramidal cells. And they are worse in the combination of pattern separation and energy efficiency, which is reassuring, right? That those models are worse because the theory says that they don't care about pattern separation that much. Okay, so I would like to summarize that the experimental derived granule cell model can perform pattern separation in line with experimental data. Pattern separation can be costly, and granule cells seem to be Pareto optimal for the combination of pattern separation and energy efficiency. And I think I didn't show this to you, or I went very, very fast through it. But it's also interesting for adult-born granule cell specialists that they seem to be more energetically efficient, but then they are worse in pattern separation. And it looks like in the development, the immature granule cells, they become mature granule cells, but they seem to move along the parental front, which is very nice. At least that's what our models indicate. Okay, I think I am at the end of my talk. I still had some outlook, so you can do this at the circuit level, but I will skip that. And I still had one more part, but I think I will not talk about it because this was another example for Pareto optimality, but in the visual cortex. So I think I will just skip that, and I will just give you this is experimental data that we modeled, but I'll skip that. And I will just give you the reference. So if you want to read about Pareto optimality in the visual cortex, in visual cortex neurons, then you can look up our new preprint in which we explored these ideas of Pareto optimality and degeneracy. So, okay, I will just skip that and I will come to the end. So basically that's it. And I am arguing that real neurons are moving along trajectories in the low-dimensional manifold in the parameter space, and they are trying to be Pareto optimal for more than one objective. One objective would be energy efficiency, and another one would be information processing. I think that would be a good summary remark. So thank you for your attention, and I'm happy to answer your questions.
Thank you, Peter. That was a wonderful journey, and